This is a production of Cornell University.
Well, thank you all for coming to the seminar today, and thanks for the invitation from Plant Biology to come here. This is really cool for me. You know, at Michigan State, we talk a lot about Cornell, and I see at least one go green there. We love sending our students to you guys, and we also love talking about you guys because, you know, we try to benchmark a lot of our programs. A lot of our faculty are Cornell grads, and we hopefully we send a good number of students your way and vice versa. And the breadth of sciences, plant sciences here at Cornell is very much within my comfort zone, similar to MSU. We have about 180 faculty that work on plants at MSU, so quite similar. We run the breadth from genes and genomes all the way to food security, food safety, and production. So again, very similar. This is a new building, just for those of you who haven't been to MSU in a while. So the idea for this building, this is a kind of a translational hub. And you'll look at my research that I'm going to talk about today. Maybe the stuff I talked with Chris Smart about is more translational. The stuff I'll talk about today, you'll say, how did you get in there? So I share a lab with Xingyang He. He's one of Alan's proteges, and Greg Howe, who works on insect interactions, but Christoph Benning, Dean Della Penna. And the idea is that this building bridges the gap between the crop and soil sciences, the plant and soil sciences, and the fundamental research in plant biology. So this is just some of the new things that we are doing here. So in large part, what I'm going to talk about today is framed by the history of my training as a postdoc and then as a graduate student, or excuse me, as a postdoc in Brian Sasowitz lab, and then as an early assistant professor. And in large part, when I arrived at MSU, you know, I was convinced that our genes and effector genes were going to solve all of the world's problems, and you know, we'd just find those magic bullets. And so we did a lot of really deep drills on understanding the basic mechanisms of how plants and pathogens communicate. And in large part, these are great tractable systems. And I'm going to talk about one or two of these today in my talk, but resistance genes from plants are recognized by specific avirulence genes or broadly effector proteins and pathogens. And it's this interaction and the function of other auxiliary proteins, chaperones, et cetera, that give yield to species level interactions in host range and resistance ultimately. And one of these, as I started working in Brian's lab, you know, we have this kind of general diagram, this bifurcation here, that once a pathogen is recognized, it signals through one of two broad pathways. One is EDS1, which Jane Parker's group has really done a remarkable job in defining the processes that function through this pathway, and a number of other labs have also in this pathway, Jen, for example. My research initiated by working on a protein called NDR1. And NDR1 is required for the activity of a specific class, a CC-NB-LRR class of proteins. And like any genetic screen, death is a great and easy phenotype to identify. So NDR1 was it's kind of our nude mouse model. You just look at, and still it's the bane of my graduate students that work on this, their existence. You look at this plant, it just gets sick. And we're working towards understanding and defining what role NDR1 plays in broader immunity. But to do that, we've really taken a different approach and asked, what is the function of NDR1 in the absence of a pathogen? And I think that is really the power of using effector proteins in this day and age with genome sequencing and transcriptome analysis, and really the ability to easily collect the diversity of pathogen isolates and their hosts from all over the world, is understanding how did critical proteins, these auxiliary proteins or these resistance and avirulence proteins, evolve from a basal function, a physiological function. And to do that, you know, 6 or 7 years ago, you had to really use a Arabidopsis. And I made a comment last night at dinner, and I think the chair of plant biology made a comment about model systems. You know, the model systems are great. A Arabidopsis is a great model. Now in this day and age, if you've got an Illumina high-seq or even a MySeq, you've got a model system. You just set your plant next to it, sequence it, and it becomes a model system. So one of the scariest realizations that I had in talking with Xingyang a few years ago, I said, Xingyang, do you know how many people work on Arabidopsis at MSU? And there are actually 9 of us, 9 out of the 180. And many of the people, you know, now are working on, potatoes are really important field of study at MSU, tomato. I do some work in cucumber, but all of these systems have quickly become model systems, and more importantly, I think they're important ecosystems to understand. They're important for food security and ultimately for food health. Now, using effector proteins, I'm obligated to show this slide, so I apologize in advance. I have to show a slide like this, but this will be the last and only one. Using these effector proteins, we can probe immune systems. We can probe, human systems, far fewer effectors in human cells, but they can use these to define the physiological processes that not only regulate immunity, but they can really define the regulation of these in the absence of pathogens. And we do the same types of things with plant pathogens. So, you've got ubiquitination processes. AVR-PTO is one of the, if you don't know AVR-PTO, maybe this is a seminar down the hall that you should be going into. Greg Martin, Alan Comer's groups, they've really, all of this DC3000 effectors. But the point of this slide is really just to illustrate the breadth of targeting and the breadth of processes that are involved in immunity. Immunity is not just R genes, it's not just A virulence genes. And so what my lab has done is we've taken a step back and said, let's forget about the R genes. They're great, they're tractable systems, but let's focus on the physiology. So what my lab has been focusing on is a couple of things. One is, because we are a land-grant university at Michigan State, and there are a number of crops, we started working on using a functional genomics approach to understand cucumber downy mildew interactions. So, and this is, I used to giggle a little bit when I'd say this, but if you eat a pickle east of Denver, Colorado, that pickle came from Michigan. We're the #1 producer of pickles. So at a land-grant university, when you've got these partnerships with industries, it's quite easy to get industry funding to work on an important problem. And so we started working on this, but we totally, we took the gene jockey-based approach. We took it out of the field. We started developing the resources so that we could deploy these. And we've made some marked progress in this area. One recent paper I want to just point out, just came out a few months ago, a week ago, is on a genome-wide survey of alternative splicing. And so this really kind of represents a new area of interest in my lab, is asking how pathogens can reprogram their host, and in turn how a host can respond at a transcriptional level to develop and evolve resistance responses, and in turn the pathogen can evolve virulence responses. So we also work on, obviously, plant pathogen interactions, and I don't know if it was Alan that has said this to me before, but NDR1 is really kind of an enigma and a black box. We don't know what this protein does. And I'm going to show a picture here that I think we've got at least a snarky idea of what it does. And then what I'm going to talk largely about today is the function of actin cytoskeleton. And actin is, as I mentioned, is broadly required for a number of host processes, and it is targeted by pathogens in both plants and animals. But the real key of understanding and really kind of bolstering my interest in focusing and shifting the focus of the lab to actin was an observation that we made while working on NDR1, and we said, well, let's see what the actin cytoskeleton looks like. And it actually, this is evidence that my graduate students will agree, NDR1 is absolute evil. It looks like, for those of you who are not absolute nerds, this is from a popular movie. I won't illustrate my nerddom here. But this is what the actin cytoskeleton looks like in an NDR1 mutant. This is a wild-type Columbia stomata. So this is going to be kind of the focus of my talk today, is kind of defining how immunity regulates actin cytoskeleton dynamics and how pathogens have evolved to target this process. So today, I'm going to give you just a brief introduction to the actin-pathogen connection. I'm going to talk just a couple of slides about some published work, but most of what I'm going to show is some new and unpublished data. So I'd welcome comments and opportunities to kind of, entertain the idea, Brad, you're crazy, or, oh, wow, this is really neat. I'm going to talk a little bit about effector targeting, and we've done a genetic screen and with help from a postdoc in Xingyang's lab, Brian Kvitko, who was a graduate student with Alan, and we've identified 2 effectors that specifically target the actin cytoskeleton with opposing activities, much like what's observed in salmonella interactions with mammalians. And then finally down to the regulation of this process. And this will get us into another effector protein that specifically targets a calcium-dependent protein kinase. So again, what I'm going to show here is largely grounded in the unifying theme of quantitative imaging at the plant pathogen interface and some new techniques. So broadly, the actin cytoskeleton is really, it's cool. I mean, that's what the cover of our journals, are sometimes littered with. They just these really sexy pictures of trichomes, and the actin cytoskeleton is involved in determining cell shape. is involved in movement. It's involved in processes such as gene expression, growth, pollen tube development, stomata open and closing, not only during pathogen infection, but during the diurnal cycles. And work from Jens Stugard. This was about 7 or 8 years ago. Really one of the first images, these are static images, but showed that the actin cytoskeleton was, they could somewhat quantitatively assess the rearrangement of the actin cytoskeleton in response to a microbial infection. So this is a rhizobium, this is a Lotus japonicus root here, and you can see that during the infection process, you go from these transverse arrangements of the actin cytoskeleton more towards these longitudinal processes. And in large part, that mediates the growth, the orientation, the development of the root hair, which ultimately lead to nodule formation. So as a platform for pathogens, arguably some of the best work has come from the work of Listeria and other pathogens, Yersinia pestis, for example, which demonstrate a couple of mechanisms. So in Yersinia pestis, the pathogen targets actin cytoskeleton somewhat indirectly by shutting down phagocytosis. Listeria, I think this is just really cool. Listeria actually expresses actin binding proteins, actin polymerizing binding proteins on its cell surface, and can actually usurp the actin cytoskeleton of its host and create these actin rockets, which it uses to propel through the cell. And it's just fascinating. On plant pathogens, a little bit less exciting, less cool-looking pictures. But nonetheless, the actin cytoskeleton has been demonstrated. So work from Ralph Panstruga's group showed that you can get rapid reorganization of the actin cytoskeleton to the site of penetration. And it's believed that this is not only as part of the callus deposition response, but could also be involved in trafficking, not only proteins, but organelles to the site of infection. So you can think about this kind of like the fort that's being attacked by the bad guys who are under the control of the Eye of Sauron. Right, you're going to reinforce your front door, reinforce your wall, and you need to bring all of your resources there. In Pseudomonas syringae, not a lot is known about this. We've published half a dozen or so papers, and some in collaboration with Chris Steiger's lab, to actually ask, how does Pseudomonas syringae manipulate host actin cytoskeleton? And remember that Pseudomonas remains extracellular during its infection process. So the logical explanation is it must be doing this either one through secretion of a toxin or manipulation of some sort of extracellular matrix component, or through effectors. And so we kind of took a little bit of approach and asked, well, is there a PTI component? Is there a cellular, extracellular component such as flagellin that manipulates actin? But is there also a secreted component that can manipulate actin? So how do we do this? The static images are great, but I made a comment this morning that microscopy is one of these things that, to be honest, you can see what you want to see. You can look through the microscope and say, oh, wow, there is some different looking stuff there, and I'm going to snap that image and do it 5-10 times, and you're done. We really wanted to step away from that and to assign, really let the computer do this for us. So what we did was we collaborated initially with Chris Steiger at Purdue, who had developed the turf microscopy-based method and adapted it for plant pathogens to monitor actin cytoskeleton. So as an organelle, as a structure, the actin cytoskeleton is really dynamic. It undergoes this continuous cycle of phosphorylation, of depolymerization and polymerization. And this is a very rapid, this is even slowed down. These filaments will grow at micrometers per second, and they're redistributed. And they're redistributed in a stimulus-dependent manner. We still don't know the endpoints, how an actin filament may be anchored to one portion of the cell or not. But what we did was we said, well, let's see if we can use this method of monitoring, this TIRF microscopy, which stands for Total Internal Reflective Fluorescence Microscopy, and take angle of incidence measurements of the actin cytoskeleton during infection. And just our first simplest observation here, you can see that there are striking, and I'll show you even more striking images. When you inoculate a plant expressing tagged or marked with GFP for the actin cytoskeleton, you can see a mixture of dense arrays of filaments. These are just fine filamentous structures. And then you can see these periodic alignment of these of bundled microfilaments. But when you inoculate it with a virulent strain, you can see the disappearance of these this dense bundle array, and most of it goes into these highly arranged bundles of actin filaments. I'll ask this question now, and then I'll ask it again later. We still don't know, though, is this a cause and effect? Is this a part of the defense response, or is this part of the virulence mechanism? And so this is where a lot of our work using some of the strains that we got from Alan's lab has come in really handy to enable us to really ask the question, are we looking at a virulence question here, or are we looking at a host immune question? And a lot of this started with work from a former postdoc in the lab, and this is one of these dreams that I wish upon any young assistant professor, is that I got kind of like the best postdoc ever. She was a graduate student in Sophia Camus' lab, and then she was a postdoc here in Dan Klassig's lab, and her husband had a job at Michigan, so it was one of these things, okay, well, let's go to Michigan State, and she just came in and published 3 papers in like 3 years and did a great job, and then ultimately came back to Dan's lab 2 more years, and now she's got the dream job in Hawaii. So there are still jobs out there. You may have to move to Hawaii to get them, but that's just for all you young people. So Miao-Ying did a screen. So we got the idea, said, well, let's think about actin. And she said, well, let's look at what's known about actin in the mammalian systems. And Yersinia pestis secretes a suite of 5 or 6 YOPs. And these YOPs stands for Yersinia outer protein. They're secreted effector proteins. And they target Rho GTPases, and they shut down actin. So is there anything in Pseudomonas syringae that might be similar to YOPs? And so she screened what was at the time some of the available Salk lines available from the Resource Center, and screened it against all of these Pseudomonas syringae expressing effectors. Here's NDR1 over here, kind of our nude mouse model control, sick in all cases. And what she identified was in this ADF4 mutant, AVR-PPHB was led to a susceptible interaction. So really nice, quick, easy genetic screen. From there, we moved on and really characterized, that was all published 6 years ago now. We've been focusing a little bit more on actin depolymerization factors. In Arabidopsis, there are 11 of these actin depolymerization factors, many more in plants than there are in humans, many more actin isoforms in plants than there are in humans. So we've kind of developing this idea is that is actin, because of the number of isoforms, is it highly specialized in plants? Does actin 7, for example, perform an immune role in plants? In humans, there are only 2 actin isoforms. Plants, like I said, have about 12, 10 are expressed. ADFs in general, they bind filamentous F actin. They break it in half, they sever it, and then they depolymerize it into monomeric G actin. And so we focused on this. We wrote a proposal with Chris Steiger and Jeff Chang, and it was funded. And the basic idea of this proposal was we were going to poke every actin-binding protein in Arabidopsis against Pseudomonas syringae, and then repoke it again against all of the effector proteins. And so it comes out to about a combination of 2,100 or so pokes that we were going to make. And the basic idea was to understand not only the immune function, but the regulation of this process. So F-actin is depolymerized by ADFs, broken down, it's recharged by a capping protein, ATP loaded, becomes a substrate for profilin, and it's added on to the minus end here. So it's this repetitive, continuous cycle. And we had begun screening some of these for phenotypes, and we had found that if we start to perturb this cycle, if we kind of speed it up or slow it down, we could speed it up potentially by knocking out an ADF protein, so then you don't have as many substrates acting upon your F-actin, so you slow down the conversion from F to G. Or we could speed it up by overexpressing actin and just increasing the actin pool. One important point is in plants, only 5% of actin is polymerized. In humans, it's about 95%. So plants have this huge reservoir of unpolymerized actin, and from a biochemical standpoint, you could hypothesize that this would support explosive rates of polymerization, right? You highly regulate this population, and when you need it, you thrust it into the cycle here through the activity of profilin. And so that's why you can see, these dynamic polymerization and rapid depolymerization processes I showed you a few slides ago. So we got the grant. We followed this up quite quickly and followed up Miao-Ying's work by showing that ADF4 is required for effector-triggered immunity. Miao-Ying had already demonstrated that, but we really wanted to ask, what is the mechanism? And so what we identified was that ADF is actually required for the expression of RPS5. And again, all of this was published a few years ago. Phosphorylation is highly diminished in an ADF4 mutant in the presence of the effector AVR-PPHB. I'll apologize for all the alphabet soup for those of you that aren't used to it. So AVR-PPHB appears to at least target MAP kinase signaling in an ADF4 dependent manner. And kind of just some of the simple biology here, really demonstrating that phosphorylation and the activity of ADF4 is phosphoregulated. ADF4 phosphorylation leads to a dissociation from actin. And that's just the basic concept of, it's kind of like doing a kinase assay to demonstrate that a kinase is a kinase. This just demonstrates that an ADF is an ADF because it binds actin and is phosphoregulated. But this phosphoregulation has an immune function, demonstrating that when ADF4 is unphosphorylatable, the plants are sick, and this is also revealed by the induction of the hypersensitive response. So in a wild-type plant, AVR-PPHB induces an HR. When ADF4 cannot be phosphorylated, you don't get an HR, and that ultimately leads to disease. So that really got us into kind of the first deep drill, and I'll talk in a few slides about how we're furthering this. So the important take-home here is that ADF4 is regulated by a kinase, and this kinase appears to be associated through some virulence activity of AVR-PPHB. And in a few slides, I'm going to show you what that kinase is and hopefully provide some evidence that there is a specific interaction. The next year, we published that there was actually ADF4 response to PAMPs. This was work we collaborated with Chris Steiger's lab on, and showed that there are actually two responses in plants in response to pathogen infection. There's a very early response that occurs between 1 and 3 hours, where you get this burst of actin filament organization. And this was largely attributed to the activity of flagellin and chitin and EF2, other pathogen-associated molecular patterns. And then this later response that I'm going to show a little bit more in detail, some more unpublished data, that occurs around 24 to 30 hours, and this later response. So we really, we've got this temporal separation, which worked out well for us. We can look at early events that we can genetically determine and define as being associated with the function and activity of PAMP recognition. So we've got all of these mutants. You can knock out the plants, and if you can knock out this early response. And then the big question, though, is what is the pathogen doing to prevent this? So let me back up. This early response, we believe, is immune related. We believe that this early response obviously is required, and we've done all the growth curves. This early response is kind of priming the immune system. It is the kind of the deep breath the plant takes when the pathogen first initiates its infection. And then this later response, we believe, is a virulence response. It's a virulence mechanism. This lag period is the plant and pathogen first shake hands, then they do this little dance for a while, and then if the pathogen is going to win, it's going to come back in with effector proteins and further change the actin cytoskeleton, actually change it in an opposite way. So this is a bundling or density response. So the plant starts making a lot of actin at this early response. Again, this kind of deep breath. And then at this later response, the pathogen comes in and just bundles the actin cytoskeleton. And what that does is that diminishes the plant response, the plant ability to have this dynamic structure that's involved in immunity. So the question was, are there Pseudomonas syringae effectors that dampen or stimulate changes in the actin? And can we identify these? And do they, ultimately, just by definition, are these effectors functioning to block PTI responses? And is it an actin-based PTI response? So to address this, we really started getting into really fine details, and we asked the question, okay, we've got to develop a new system of how to measure changes in actin. A really highly quantitative, highly unguided. We didn't want to be looking at images. We didn't want to be looking at 900 images, which is what some of these. So for each one of these experiments, we collect about 900 images of each time point, of each data point, and then we feed them into a computer, and the computer tells us, yes, this is significant. But we collaborated with a group at the University of Tokyo that took it one step further, and they actually developed an algorithm that could identify within this population of actin, within a single cell, 4 different organizationally distinct actin structures. So you've got filamentous, you've got longitudinally bundled, you've got radial bundled, and then you've got this dense microarray. And they developed an algorithm that could go in. We take all of these images, we feed them into ImageJ, do a hierarchical clustering, and basically it spits out these 4 classifications. So with this, what we started asking, and I'm just going to show you one slide to show you where we're taking this, is, can we use physiology and compare that to immunity. So we all know that stomata open and close during the diurnal cycle. And to the untrained eye, an open stomata looks the same under light conditions as it might look under a 6-hour pathogen infection. But in fact, they're quite different. And a couple of other observations that we've identified is that the pathogen infection actually introduces a transient deformation of the stomata, kind of bounce back the red highlights there, compared to open and closed under diurnal cycles. When you inoculate these with Pseudomonas syringae, you get this deformation. It's probably kind of some turgor pressure on the adjacent cell as a result of the infection. But then it recovers. But you can see that an open stomata by a pathogen looks markedly different than an open stomata by light cycles. Highly bundled, highly dense radial transverse arrays, as opposed to here more of a mesh network of radials. So that's, we can quantify this. So the big question was, can we go in and use these types of analyses to identify specific effectors using plant genetics and bacterial genetics to identify actin cytoskeleton? Can we measure these? And can we separate cause from effect? Can we separate the immune response from the pathogen virulence response? So just two definitions I want you to remember for the next 5 or 6 slides. Whenever I say skewness, this is really, it's a mathematical measurement. So we're looking at the distribution of actin. It has a bell-shaped distribution under normal conditions. Skewness simply means we take that bell and we skew it to one side or the other. And that would be skewness equals actin bundling. So when you see highly bundled arrays, that means you have high skewness. The opposite is highly dense arrays means unbundled. Now, I use that word not to be simplistic here. We don't know if it's depolymerized. We don't know if it's ever been polymerized. So we assigned a very kind of a general definition. We just say it's unbundled. So an image like this, we would say is highly unbundled, it's highly dense, and this would be highly skewed. So this is mock inoculated and DC3000 inoculated. Hopefully you can see the differences there. DC3000 highly bundles the host actin filaments, whereas a HARC-C mutant really doesn't change it. Quantitatively, you can do the same. We can go in. They show up better here. Highly dense arrays, but clearly something. So this became kind of our metric. This was our first phenotype that we're going to follow. Let's screen all of the plant mutants we might want to screen against all of these CU-CPB isolates that we got from the Colmer lab and say, can we define a pattern within this? Can we narrow down a specific effector? And so we got these effectors, and the first thing is to standardize our system, set up our system so that we're taking our measurements at 24 hours. We want to eliminate any bacterial density effects, so we do a growth curve at 0 hours and 24 hours. We count the bacteria at each one, and we set up the standard curve. So this is DC3000 at an initial inoculum of 0.3, 0.03, 0.01, kind of our standard curve. We benchmark all of Allen's strains against these. And then what we do is we inoculate our isolates. So for example, we would inoculate CU-CPB 5500 at the same concentration as to compare with DC3000 at a 0.01. This strain grows at an initial concentration of 0.3 as compared to wild type. So there's a bit of a fitness impact, right? So our main concern was we want to make sure that in the end, we're not looking at bacterial density as cause and effect. And then these are just the isolates you can see. I'm not going to go through this, but DC3000 has all of these isolates, and then some of these CU, let's say 5500, is basically knocked out for all of these isolates. Delta cell, it's a conserved effector locus, is present in all of these, but not in these, et cetera. So you can start to line these guys up and say which effectors. And ultimately, you get down to 2 or 3 strains that are missing the same couple of effectors, and then you can knock those out. So our inputs was pathogen genetics using the CU-CPB strains. It's defined tractable growth. We can say, okay, we know what disease looks like. Death is a really cool, easy phenotype to identify. We're going to look initially at a single host, and we're going to use the power of these overlapping effector inventories. Our outputs are going to be disease assays. So again, using skewness and density, right? So these are kind of opposite. When you have high bundling, you have low skewness. low density, and vice versa. So again, kind of these opposites, black and white, are really where you will want to be to benchmark these. And we can do this using a computer algorithm to quantify with no human input. So just quickly, I'll go through this. This is all work that's in revision. So we screened through all of these lines, and we've narrowed it down to a couple of the CuCPB isolates from Alan's lab. And we narrowed it down to about 5 effectors, actually 6 effectors. I'll tell you about one of them that we're not really following up on. We're playing around with it with Xingyang's lab a little bit. But we narrowed this one down to 5 effectors. We knew that there were one of or multiple of 5 effectors that were responsible for inducing this decreased actin response. Okay, so it came down to this isolate here, and then this one here. So like Salmonella, we identified that Pseudomonas syringae effectors have these opposing activities. Pseudomonas syringae or Salmonella has one effector that binds host actin, and they have one that bundles host actin, one that unbundles host actin. So you can imagine, I'm a very analogy person. My students always tease me. Imagine walking into this room with all the chairs are littered in different directions, and you had to walk through. You would push some chairs out of the way and take a step, then you would pull some chairs. So this is what the pathogen is doing. It is pushing and pulling on the actin cytoskeleton depending on the state of the immune signaling pathway. I'm going to talk more about this one. For those of you that are curious, AVRE actually induces actin polymerization in its host. And we don't know why. We're working with Xingyang He's group a little bit on AVRE. AVRE is one of the conserved effector locus, highly important, widely abundant, but it has a very strong effect. When you express it in plants, either transgenically in plants or through effector delivery through the pathogen, it immediately bundles the actin cytoskeleton. So we narrowed it down from this isolate CuCPB549 to this effector. This is the cluster 9. There are, 6 or so effectors on this one. We went through and we screened all of these looking at growth curves. There was no real impact of growth when we knocked out any of these individual effectors, you can see. So that really was a nice thing, because now we can do standardized assays where we're inoculating all at the same concentrations, counting all of the same growth, et cetera. Then we start to add back in individual effectors, and the long story short is that it's HOPG1 actually induces actin bundling in its host. And this was an effector that's been worked on a little bit by another Colmer alumni, Jim Alfano, that showed that HOPG1 can actually induce cell death in its host. So, and interestingly, I'm not going to show this. It's published, and we're working on getting some really a better repeat. The published result's real. We're just having trouble getting it to work the way to the reviewer's satisfaction. HOPG1 appears to bind kinesin, which is a mitochondrial localized protein. Kinesins are motor proteins that mediate the interaction between myosin and actin. And so what we believe our method is, or our mechanism is, that HOPG1 is delivered in the host cell. It immediately targets kinesin in the mitochondria, and that induces actin bundling. Actin bundling in the mitochondria, if you PubMed search that, boom, it's cell death. So that's one of the cool things about actin cytoskeleton. Depending on the distribution of actin within the cell, as well as its organization, you can tickle certain responses. If you send actin into the mitochondria in a human cell, it initiates apoptosis. If you bundle actin in plant cells and pollen cells, it induces cell death, apoptosis. So we believe that the virulence, at least maybe one of the virulence targets of HOPG1, is kinesin, and it requires a functional actin cytoskeleton. It manipulates actin cytoskeletal dynamics to initiate cell death. And we have some data that demonstrates this. So DC3000 at 4 days induces this nice chlorotic phenotype. If you knock out HOPG1, you get no chlorotic phenotype. If we add back HOPG1, it's not as strong. You recover much of that. We've done chlorophyll analysis. We recover about 70% of that. Brian Kavitko is helping us on this. We think either it's a contribution to the CMAL locus. We think we've eliminated the contribution there. Or we think it's a position effect, that we're just not getting strong expression of this HOPG1. But to demonstrate that this is an actin-dependent response, you can take DC3000, you get this yellowing phenotype. Again, a little bit clearer here. You knock out the HOPG1, the delta 9 cluster, you remove that. But if you add back in a depolymerizing factor, you can restore the cell death phenotype. This experiment by itself has been done several times, but demonstrating that HOPG1 can really compensate or is additive to this process. So just to tie up this first part of my talk is really what we've demonstrated here is that pathogens modulate the host actin cytoskeleton, and that there are multiple points of this interaction. And really, I think this is the first example of a pathogen-induced modulation of transcription back to the ADF story. We've got a link there. So the big question for us, and I'm going to wrap up my talk with this last story, was back to this story here that Katie Porter had published. So we know that ADF4 is required for the expression of RPS5. We know that AVR-PPHB is the effector protein that tickles RPS5. We also know from other work from Roger Ennis's group, from Jean-Mingeau's group, that AVR-PPHB targets multiple proteins in its host. And we've got this kinase link. Remember, the function of ADF4 is kinase dependent. So we asked, again, another one of these, I wish I had a time machine. I'd send the postdoc to Vegas, because she really had a good guess and it worked out. She said, can we identify the kinase that phosphorylates ADF4 and is, in fact, is this kinase targeted by AVR-PPHB? Again, we're hoping that maybe we can identify the virulence target. And thanks to Greg Martin and his lab for publishing this just a couple weeks ago, because I was looking for a slide that demonstrated the function of kinases in plant pathogen interactions, and these are the two that I wanted to show. So AVR-PTO is one of the best characterized kinase signaling mechanisms through PRF and PTO kinase. Roger Ennis, I'd mentioned this is what I'm going to focus on. AVR-PPHB is a cysteine protease that cleaves the kinase PBS1 that leads to the activation of resistance. Jean-Mingeau's group demonstrated that not only does PPHB cleave this kinase, it cleaves about 20 others. So about 20 other PBS1-like proteins. Many of these are functionally unknown, but some of them have been identified. Some of them are required for the function of FLS2, such as BAC1. And so postdoc in my lab, Iru, said, well, let's set up this type of process. We hypothesized that the pathogen delivers effectors. These effectors target a kinase, and this kinase is required for the phosphorylation of actin, which leads to immunity. So we took this template that Jean-Mingeau's lab used to screen other potential targets. And again, it was a bit of a naive approach. We said, oh, well, let's just look for the 3 amino acid target sites that AVR-PPHB cleaved. And Jean-Mingeau's group had demonstrated BIC1. Previous work by Roger Ennis's group and Jack Dixon's group showed that PBS1. There are some other sites up here, but we said, well, just forget those. Let's just throw this at it really quick. And what Iru identified was the CDPKs, calcium-dependent protein kinases. Also, some of these have this cleavage site. So the question is are there additional sites? Is there recognition of this site by the kinase, or excuse me, by AVR-PPHB, and can it cleave? Big question is, are there additional substrates? Was Jean-Mingeau's work saturating? And are any of these substrates the candidate that phosphorylates ADF4? So this is basically it. We're going to look for the effector that targets the kinase. And what Iru found was CPK3 actually does both. CPK3 phosphorylates. Shoot, I forgot to put the phosphorylation. Trust me. Famous last words. CPK3 phosphorylates ADF4, but more importantly, from a plant pathology standpoint, CPK3 actually is cleaved by AVR-PPHB. So this is the experiment here. If you include the buffer only, you see nice expression of AVR-PPHB in the presence of, CPK. In the presence of AVR-PPHB, you get cleavage. If we mutate this cleavage site, we don't get cleavage. If we include a cysteine to serine mutation, it's a catalytic mutant. So AVR-PPHB is a cysteine protease. It's a catalytic triad, a histine to serine, and a histine, serine, and a cysteine. If you knock out one of these, we significantly reduce the cleavage. We're getting a little bit of cleavage here, we believe. And if you knock out both, again, you block cleavage. The real proof is in the pudding. If you knock out CPK, what does it do from a disease resistance standpoint? And as we would have predicted, if we knock out CPK3, these plants are susceptible when inoculated with AVR-PPHB. the reciprocal. We've done this about 50 or 60 leaves. No HR when we inoculate the plants with AVR PPHB, whereas with Columbia you get an HR. None of these plants appear to be affected in the hypersensitive response. What's really cool is if we add in an actin modifying agent, co-infiltrate it, we restore the hypersensitive response to wild type, kind of demonstrating that there is a functional role for actin in this process. And then this is just last week type data, but just looking at the function of the kinase as a relationship to actin, you can see that if you knock out the kinase and mark the line with actin, you get this really high density actin array indicative of an immune type response. So we're hypothesizing that CPK3 is required for immunity to Pseudomonas syringae, and that actin is a big part of this process. And this is, again, just quantitative analysis, but I think that the difference here is striking. This is only 90 images, so we've got about 800 to go to get this, but it's looking pretty good. So just to wrap up, we're really focusing on this process here of how actin cytoskeleton touches many processes. Immunity, again, is a great phenotype to study, especially when death is involved. We know that CPK3 phosphorylates ADF4, and this phosphorylation we demonstrated in a previous paper is what kicks ADF off of actin cytoskeleton. So that eliminates breaking down or depolymerization of actin through phosphorylation. AVR PPHB cleaves CPK3. We're doing the in vitro experiments now where we do recombinant express of each of the proteins, and then we'll send it for mass spec to definitively define that cleavage point, that cleavage site. We know that CPK3 is required for wild type actin filament organization. So we believe that actin is, or at least CPK3, from an actin standpoint, ADFs are the key regulators. I mean, I drew it just like we all draw the globe. Whenever you see a globe that's printed in the United States is right there in the center, and it's like the biggest country. We believe from the standpoint of actin, CPK3 and ADFs are like the players here, and CPK3 therefore would represent a key regulator of actin filament dynamics. And we know that CPK is required for resistance. And CPKs have been previously identified as required for stomata opening and closing. So we think we're on the right trail here. Julian Schroeder's lab published a few years ago in PLOS Biology that if you knock out CPK3, you diminish the ABA closure of stomata. And for us, at least, it makes a theoretical, logical step to say that pathogens entering through stomata would require or potentially target CPK3 to eliminate stomata closure. So the big picture of where my lab is primarily interested in moving is defining actin cytoskeleton as kind of the ideal surveillance platform. One of the neatest observations I think we saw, and I'm trying to figure out this experiment of how to do it in plants. But if you talk about actin, and this is a review we wrote a few years ago, it's a very highly regulated process. There's about 75 actin binding proteins in plants. There's about 250 in humans. So it's a tightly regulated process. But if you push actin into the mitochondria, you initiate death. That's true in animal systems. At least, we may have some signal with the HOPG1 kinase and actin relationship to the mitochondria that we may have a similar type of process. So our hypothesis is really that actin is the ideal surveillance platform. It touches many, many processes. It touches physiological processes such as stomata, gene expression, organelle movement and organization, as well as immune processes such as internalization of receptor complexes, vacuolar processes, immune signaling processes such as the expression of RPS5, CPK3, et cetera. So our big question here is, can we alter, not necessarily the dynamics of the actin cytoskeleton by knocking out an actin binding protein or overexpressing an actin binding protein, but can we alter a cell's response by simply changing where actin is localized? And we've started to mess around a little bit with these. We've got some inducible lines and some stable lines. But basically, can we thrust ADF4 into the nucleus almost exclusively by putting strong nuclear localization signals? And the preliminary data here is that when we do this, plants don't like it. But they really don't like it from a plant pathology standpoint, from a plant pathogen standpoint. These plants are highly susceptible. We've altered this dynamic organization of the actin cytoskeleton. So that gets back to the overarching hypothesis that we have, is that in humans there is cofilin and there's ADF. There is beta-actin, and there's one other actin. So there are 2 ADF proteins in humans, 2 actins in humans. In plants, there are 12 actin isoforms, there are 11 ADFs. Are there specific partners and pairs of these that are required for very specific responses? And I was glad to see Gene Greenberg published a small little paper in a little highly specialized journal recently that suggests that their work, where they've identified HOPW1, you may know this as HOPX, Alan, from the meeting a few years ago, they demonstrated that Actin 7 is required for immune signaling. And whenever we do immunoprecipitation experiments, we always pull down Actin 7 and Actin 2. So we think there are certain partners, kind of a lock and key mechanism, where ADF4 may preferentially depolymerize Actin 2 or Actin 7 filaments, and that this is the immune signaling specific pathway. We have no mechanistic support for this. It's simply a hypothesis, and unfortunately, a lot of the evidence out there would argue against it, but all of the evidence out there is human-based, where there's only 2 actins and 2 ADFs, so they don't have the power of numbers. So with that, I'd like to just thank the people. This is a relatively recent lab picture. Brian, I was telling Alan, Brian's kind of many of you may know Brian. He's really extroverted, a lot like me. So anytime there's a camera and a picture, he kind of inserts himself into it. So he's an honorary member. This was celebrating one of my postdocs' 40th birthday, and so we all got shirts that say "Obey Masaki." So essentially, this is the lab. So Iru is the one that has identified the CPK. Katie graduated in December, and she was the ADF person. Alex is a new student who actually will be spending 3 months in Japan this summer to do more of the actin stuff at Nagoya University. Masaki is the one that has worked not only on the HOPG1 story that's in revision, but also is doing this very precise measurement with Tokyo University on the stomata organization. And the other people, NDR1 and NDR1. And this is my lone cucumber student who's graduating and moving on to really great things. So with that, I'd like to be happy to take any questions. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.